Okay, Agnes, take it away.
Yeah, perfect. Well, thanks everyone for joining, and thanks specifically to Carlos, who's our speaker today, and yeah, who accepted our invitation. That was pretty short notice. I think I only contacted him in October, so thanks a lot, Carlos. Uh, Carlos is going to talk about seeds and seedling ecology of Melastomataceae, a chapter that was also published in the Melastomataceae book that many of you have read or contributed to. So I guess you can go read up if some things go too quick. Carlos is currently doing his PhD at the University of Minas Gerais in Brazil. He's originally from Colombia, so coming from a country with a lot of melastomes, going to a country with a lot of melastomes. So I'm pretty sure he has a lot of very exciting details to share with us. And yeah, without much further ado, we haven't heard much about seeds or seedlings here at all. So yeah, Carlos, please take the floor. We are very excited to have a new topic covered.
Okay, let me share with you my screen. Hopefully it works well. And you should be seeing my screen now.
Yes,
And now it should be on presentation mode.
it is. Perfect. Thank you.
Perfect. Well, hello everyone. I hope you are doing well and having a nice week so far, wherever you are. And as Agnes already said, in this presentation, I'm going to talk to you about the seed germination ecology of Neotropical Melastomataceae. Most of this presentation is heavily based on the book chapter that we wrote a couple of years ago, but hopefully I will try to summarize everything, mostly everything of what we wrote for that chapter. And in this presentation, we are trying to look at the history of seed germination ecology in this family by looking at these different stages, the past, the present, and the future. So we will start by briefly talking about the role of melastomes in the history of seed germination ecology in the tropics, and then ask ourselves where are we and which are the gaps regarding the research in seed germination ecology for this plant family. And knowing this, knowing the state of the art of seed germination ecology in Melastomataceae, we propose some research avenues that we should start to follow if we want to fill all these gaps that we identified in our review. So, talking about the past, the history of melastomes in seed germination ecology in the tropics, I want to believe that melastomes are at the heart of germination ecology in the tropics, either because in the first studies around the decade of the 80s, you always have a melastome in all these classic and seminal studies about germination ecology, especially because most of melastomes are part of this commonly studied group that are pioneer plants. So every study about the germination ecology of pioneer plants always have a melastome. So we always see this in these pioneer studies about the germination ecology of tropical species. And a couple of years later, we start seeing different community scale studies around the germination ecology of tropical ecosystems that included different melastome species. For instance, one of the seminal studies led by Aaron Ellison in 1993 was pretty important for the history of the study of germination ecology in not only Melastomataceae, but for tropical ecosystem as a whole, because this is one of the first studies that tried to use a comparative approach to compare species from different, if I am right, this study have around 30 species, different species from different microhabitats, from different families, from different genera, and all the logic and the rationale of this paper was to try to compare the differences in the germination behavior of species from different growth forms, microhabitats, and these kind of things. So this was the first study that actually illustrated the diversity of germination behaviors in the Melastomataceae. And 3 decades has already passed since the study by Ellison and collaborators, and we see that melastomes are always present in the different reviews and syntheses about the germination ecology of these different biomes in the tropics. And for instance, we do have a relative recent review in 2013 about the germination ecophysiology of Melastomataceae in the Cerrado and the Atlantic forest. But still, we already lack a comprehensive synthesis of the germination ecology of these species in the whole neotropics. So precisely for this book chapter that will lead us to the present of the study of germination ecology in Melastomataceae, we aim to provide an updated and expanded synthesis on the germination biology of Neotropical Melastomataceae, and we focus in 6 specific topics, which were seed viability, seed longevity, soil seed banks, seed dormancy, seed germination, and biotic interactions. And surprisingly, we found 111 studies carried with 192 species from various genera and almost, if I am right, almost all tribes of Melastomataceae. And while this number, for me, germination ecology seems like a huge number, you don't have families with this lot of studies. But when you put these numbers in perspective, considering the high diversity of species that there are of Melastomataceae in the neotropics, we see that only around 5% of neotropical Melastomataceae species have at least one study on its germination ecology. So we actually see that considering the high diversity of this family, we don't have that many germination studies for them. And when we start to evaluate which species, which genera, we start to see some interesting trends. For example, we see that the genera with most of the studies are Myconia, with around more than half of the species studied for the whole neotropics are from the genera Myconia, and only a small fraction of all other genera, such as Pleroma, Myclorrhiza, have a small amount of studies. And interestingly, we were able to find that at least that there was a study for no, sorry, this is a confusing phrase. Every tribe has at least one study about germination ecology. So at least we know something about the germination ecology of each tribe. But if we start to look at the diversity within each tribe, we see that no more than 17% of the species within a tribe have at least one germination ecology study. So we do really have a huge phylogenetic gap considering the high diversity of this family.
Eso lo bajó Paulina.
So yeah, that we have a huge phylogenetic gap, as you can see. And when we start to classify these different germination ecology studies, we see that nearly half of the studies that have been carried up to date are mostly lab experiments around the effect of abiotic factors on germination, especially, as you can see, light and temperature. So we do have a huge thematical gap in the sense that most of our studies are concentrated in a specific area of the germination ecology. Other studies, for example, around the seed longevity or seed dormancy or the interactions between seeds and other organisms are relatively scarce. So we do have a interesting gap to be fulfilled here that we are going to talk later more. And if we try to map where these studies are being carried out, we see that a huge proportion of the studies were carried in Brazil, especially in the southeastern region of Brazil, in the Atlantic forest and the Cerrado biome. So we do see that there is a tremendous gap, geographical gap, on the germination ecology of neotropical species, which tremendous gaps, for example, in the Andes, where we do know that there is a huge diversity of melastoma as well. And we only see this huge concentration of studies in Brazil, some interesting and important studies carried out in Panama, and an important number of studies also carried in the Amazon. But as a whole, we do see a huge geographical gaps in this map of studies. And if we try to locate these studies in the different tropical biomes, we do see that mostly, with the exception of only two studies, all the studies in germination ecology were carried either in rainforests, what we call here the tropical and subtropical moist broadleaf forest biome, or in tropical and subtropical grasslands or savannas. So we can, considering all these together, we need to recognize and understand that our current knowledge of the germination ecology of Neotropical Melastomataceae is highly fragmented, is biographically biased, and poorly represents the high phylogenetic diversity of the family. Considering that this is one of the most species-rich and important family that is present in all of the tropical biomes, is quite, I don't know, surprising and worrying that our knowledge is so little and so fragmented. Uh, so this is one of the biggest conclusions of our review, and we were quite surprised to find this pattern. But even if this bias exists and we are capable of recognizing that, we were able to find some interesting patterns about the different topics that we evaluated. So for example, regarding seed viability, we were able to find a high variability in terms of seed viability. So when we are thinking about seed viability, we should think of two things, either the presence of a fully developed embryo and that this embryo is viable, meaning that it is able to germinate. And when we compare the values of embryoless seeds and non-viable seeds, we see that they are all over the place. We have species that have 87% of embryoless seed that you cut the seeds and you find no embryo at all. Or you can also find that most seeds in a seed lot are non-viable. We have this value of 86%. So there is a huge variability from species that produce a lot of viable seeds to species that produce almost no viable seeds. So this variability is all over the place. And if we think what could be the response, what could be the factors that could be used to explain this variability, we probably need to go back to the location of these studies and think whether we are in a situation of resource and pollen limitation, because these huge values of empty seeds and non-viable seeds mostly come from the Campo Rupestre, an ecosystem in southeastern Brazil that develops on outcrops of quartzite or ironstone rocks. And this ecosystem is characterized by having one of the most phosphorus impoverished soils in the world. So we are already in a situation where plants are in extreme a really extreme condition in terms of nutrient content, and other studies carried in other outcrop ecosystems all over the world have already suggested that the lack of phosphorus in the soil is one of the biggest limitations for producing viable seeds. So probably species that are growing in Campo Rupestre ecosystems are producing seeds, but because of the lack of nutrients in the soil, these seeds are of really, really low quality. Another possible explanation is that species that grow in the Campo Rupestre are usually locally rare. We have small populations that are pretty far away from one or the other. So probably cross-pollination between this population is rare. And this lack of cross-pollination between populations probably leads to endogamic depression. So we see that these species are, with time and evolution, are constantly producing less and less quality seedlots. So this is another explanation for this. And the other thing is that a recent study carried in this ecosystem showed that most of the flowers of these ecosystems have a really short lifespan. So the opportunity that they have to be pollinated and produce viable seeds is actually quite short. So these are all the different hypotheses that we have to think about why these species, not only melastomes, but Campo Rupestre species as a whole, produce such low quality seedlots. And what's interesting in the campo rupestre is that we see that many species have tried to find a solution, and we can think of solution as in a metaphorical sense, meaning that a lot of species in the campo rupestre are known to produce seeds via apomixis. So we find a lot of species, especially in particular in some genera that have a lot of apomictic species. So this strategy should be a way to prevent the problems regarding endogamic depression and lack of pollen transfers and cross-pollination. And we also see lots of species with polyembryony. Some studies carried here in Brazil have found that some species can have at least 5 embryos inside their seeds. So we see that we have everything from seeds with no embryos at all, from seeds that have 5 or 2 or 3 embryos. So it's really a huge variability regarding the number of embryos that these seeds have. Yeah. If you remember from that graph regarding the different topics that we evaluated, longevity was one of the least studied topics in our review. And unfortunately, there were only 4 studies carried out regarding the longevity of these seeds, and we find a huge variability from species that are able to persist in the lab from a couple of months to species that remain viable for several years. But we really do need more studies about the longevity of these seeds because considering the high diversity and taxonomic and ecological diversity of the family, we do really need more studies about the seed longevity. But when these experiments were carried out in the field where researchers buried their seeds in the soil and monitored their viability for a long time, they found that all of the species evaluated were able to form persistent soil seed banks, which means that seeds that were buried in the field were able to persist for at least 1 year. So researchers were able to monitor these seeds for a long time, and even after a year, they were still finding viable seeds in the soil. And this result is quite interesting because when we compared studies carried about the soil seed banks in the community scale, we repeatedly found that melastomes dominate the soil seed bank in almost every ecosystem you can think of, from rainforests to savannas to campo rupestres, and every ecosystem that you can think of, there is a melastome that makes out a huge proportion of the seed bank. Such is the size of these seed banks that there is a really interesting study carried here in Brazil, in the Brazilian Cerrado, that was able to find seeds of Tibuquina stenocarpa even after 20 years of invasion by these fern species. So we see a lot of evidence that suggests that seeds are able to persist for a long time in the soil, and that's what makes them an important component of the soil seed bank, and then an important component of the natural regeneration of most tropical ecosystems. Yeah. And as we will discuss later when we talk about germination, the germination requirements of the seeds do help us explain why these seeds are able to persist for so long time in the soil. But before getting to that, there is something really interesting about the germination of melastome species, which is the dormancy. I don't know how familiar are you with germination ecology or germination biology, but seed dormancy, one of the greatest mysteries in germination biology, because dormancy is a mechanism that seeds are unable to germinate even if they are in the proper conditions for germination. And you will think why a species would like to prevent germination even if it is in the optimal conditions for germination. So dormancy is a mechanism that has evolved in different species and have different forms. And what this dormancy does is to try to fine-tune germination in the most proper conditions for germination. In the case of melastomes, we do see that most of the species don't have dormancy at all. With out of the species that we evaluated, 92 of them don't have dormant seeds at all. But we have records of 9 species, 9 unequivocal records of seed dormancy for 9 species. In this table that we prepare for our book chapter, you can see that physiological dormancy, a dormancy where the embryo has a physiological imbalance of the different hormones that control germination, it's present all over the phylogeny of melastomes, from the Marsetia to the Melastome to Meconium. All of the tribes have at least one record of physiological dormancy. And what is interesting in this case is that this was studied more deeply for the Campo Rupestre vegetation. And in this study by Silveira and collaborators in 2012, they were able to find that physiological dormancy has evolved multiple and independent times across all the melastome phylogeny. And what is most interesting about this, maybe, I don't know how large is this figure for you in your screens, but if you see the black squares represent species with records of physiological dormancy, and if you see the next column of squares, they have a X for xeric microhabitats and an M for mesic microhabitats. And you can see that most of the dormancy records, the black squares, are associated with a xeric microhabitat. So what was proposed in this paper is that physiological dormancy evolved multiple times in the melastomes of the Campo Rupestre as a result of the transition of these species to xeric microhabitats. So xeric microhabitats have a lot of evolutionary pressure to restrict germination for the most proper condition that it should be the rainy season. So this physiological mechanism avoids germination to occur in other different season that is not the rainy season. And it's pretty interesting to find that most of these evolve specifically in this ecosystem, in these microhabitats, when the evolutionary pressure to control germination is higher. And if that wasn't fascinating enough for you, we have a really interesting case of combinational dormancy. This is the species Chaetostoma armatum from also the Campo Rupestres as well. And what was found in this study is that not only these embryos have physiological dormancy, but they also have physical dormancy. Physical dormancy is probably a form of dormancy that is more common to all of you, where the seed coat is water impermeable. So even if the seed is put in a moist environment, there is some hydrophobic mechanism that impedes water to enter to the embryo and germinate. So there should be some environmental or biological mechanism that degrades the seed coat before water is able to enter and start germination. But what is interesting about this species, Chaetostoma armatum, is that this is I don't know if you are seeing my pointer, but in the L figure, this is a detail of the seed coat. And what is interesting is that in all species, except Chaetostoma armatum, physiological dormancy is achieved by the presence of macrosclerates in the seed coat. So macrosclerates, these elements of the vascular transport, they are hydrophobic and impede water entrance. But in Chaetostoma armatum, we don't have this kind of a structure. What you are seeing here in the L figure L is the presence of polyphenolic compounds that avoid the entrance of water. So it's interesting that water is able to enter to the cells in the seed coat from the right side to the center of the seed, but the presence of this polyphenolic compound, let me use the point. Can someone confirm me the pointer is now appearing? Now the pointer should be appearing, yeah? That should be easier to explain. So this is the seed coat, and this is the outside of the seed. And even if water is able to enter the seed, coat, the water remains trapped in the lumen of the seed coat cell, so it isn't able to enter the seed and the embryo and hydrate them. And this is really interesting because this was the first time that such a mechanism was described in all angiosperms. All the physical dormancy that we know so far are from a totally different mechanism. And this was the first record of water, the seed coat water impermeability in the whole order Myrtales. So this was a really fascinating discovery uh, uh, for seed germination biology as a whole. And yeah, it was a really, really fascinating discovery for all melastomes people working on melastomes. And yeah. So now that we have talked about dormancy, we should talk about the germination physiology of this species. And all neotropical, if there is something that you should know about these species and seeds, is that all neotropical melastomes so far are known to germinate better under light conditions. And even more than half of the species that we evaluated only germinated under light. So if you ever found a melastome that is germinating mostly on dark conditions, that is something strange, quite different, for instance. So if you get to know something about that, let me know, please, because this should be something pretty fascinating and different. And it is interesting that species, they do require light conditions to germinate, but some species are able to germinate even if they just receive a couple of minutes from light. This is a classic study carried here in Brazil, in the Brazilian Cerrado. In the x-axis, you see minutes of light, and in the y-axis, you see germination percentage. And for example, in Miconia albicans, you see that only 1 hour of light exposure is able to produce a germination percentage equaling is statistically equal to a lot of exposure to light. And when we go to Myconia stenostachya, we see that only 15 minutes of light are able to produce such a germination percentage. So even a really small amount of light is required for these seeds to germinate. And this widespread um, light requirement is one of the characteristics that allowed Melastomataceae species to persist for a long time in the soil. Because while they are in the soil, even if there are just a couple of centimeters inside the soil profile, when they don't receive light, they won't germinate. So if they are buried in the soil, they would need to wait. They will need to wait to be bring up to the surface to start germinating. Otherwise, they will remain in the soil for a long, long time, as we saw in those germination studies. But if we uh, start to study these light requirements with more detail, we should think also about light quality, because yeah, the other studies were more about the contrast between dark and light conditions, but these more recent studies are trying to understand the difference in light requirements. If you have gone Obviously you have gone, but if you have gone to a forest, you can perceive that the light in a gap, for example, is totally different from the gap that you are having in the understory. There you can see that there you are able to see because there is light, but when you try to decompose this light in the different wavelengths that compose them, they are not the same light, because foliage and green foliage preferably absorbs red light. So when you are in the understory, there are a lot of layers of vegetation uh, above you that are filtering the light. So the light that is present in the understory has pretty less red light and has more far red light than the gap. if you are in a gap and there is no vegetation above you, light is not being filtered, but any sort of vegetation. So you are receiving, let's call it a pure light with lot of red wavelength and pretty much not that much of far red light. So if you remember your germination physiology from that undergraduate course in plant physiology, you should remember that red light is the light that induces germination, and far red light, not so much. And all this explanation about the microhabitats and the difference in light quality is for you to look at this table. When we have this study was not only carried on melastomes, but species from the gaps and the borders and the understory of Atlantic forest here in Brazil. And what is really interesting is that the requirements of light quality in these species are different depending on the microhabitat they colonize. So species from the understory, such as Leandra reversa, Meriania glabra, they have a really small ratio of the red and far red ratio that they can germinate. You need to think this may not be such easy to understand, but if you think uh, a smaller value, you should think of a more filtered light, the light that you found in the understory, because all the red light has already been captured by the vegetation above you. So even if there is really a small red light in that environment, Leandra reversa and Meriania glabra understory species are able to germinate. But if we see species such as Clidemia viscehata and Osaea confertiflora, these are species from the edge of the forest. And you can see that these species require a higher ratio of red and far red light. So they do need more red light to be able to germinate. So it's really interesting that we can see, even if all species are require light to germinate, we can see that there are some differences on the light quality that they do need to germinate. So, but studies about the effect of light quality are rare and are only starting more recently, and they are hopefully difficult to make. I'm making these experiments, and yeah, I understand why no one does that, but if, in any case. But yeah, it's interesting to find that we can see some small separations in these groups, and depending on the microhabitat, they should have different light quality requirements. And for the second uh, germination factor that was one of the most studies, we see that melastomes usually are able to germinate on a broad temperature range. If we can think of melastomes are colonizing a huge variety of habitats and ecosystems and microsites. So we should expect them to germinate on a broad temperature range. And if we compare our most studied biomes, the savannas and the rainforest, we see a sort of difference between them. In savannas, we see that the temperature range of melastomes goes from 15 degrees to 40 degrees, while in the rainforest, that are not as high, we see that they are not able to germinate in such high temperatures such as savannas. But if you start to look at the differences inside these ecosystems, they are also all over the place. This is a small example from species from the Campo Rupestre, and you can see that while all species are able to germinate between 15 and 30 degrees, the optimal germination of most of them is either between 15 or 20, or 25 degrees, and so on. So we see that the variation is quite species specific. The temperature requirements are quite species specific. And unfortunately, as we will see in the research gaps, we do need a more comparative and formal approach to compare the temperature requirements of these species. So far, our comparisons were extremely qualitative, seeing that this kind of approach when comparing which temperature does this species germinate better and so on, but we do lack a more formal and statistical approach to compare these germination requirements. And finally, and unfortunately, the biotic interactions between melastome seeds and other organisms was one of the least studied topics in our review, but even with a small amount of studies, we were able to find that melastome seeds interact with a wide diversity of organisms. In the case of predispersal interactions, we do see that melastome seeds interact with a huge variety of organisms, from galling insects that parasite fruits and ovules, from lepidopterans that predate fruits and seeds, beetles that consume seeds while they are already forming inside the fruit, ants, nematodes, and so on. We have a huge, huge community of organisms interacting with melastomes. Um, when we go to the post-dispersal situation, when seeds have already been dispersed, we do see that they also interact with ants and beetles and fungi in the soil. But unfortunately, these studies are quite scarce, are quite concentrated in a few geographical areas. So even if we are able to recognize a huge diversity of organisms interacting with melastome seeds, we actually don't know much about the relevance of these interactions in a whole community scale. And having said that, we should talk about our recommendation, our research agenda, or what should we be doing next to advance our knowledge, is that we definitely need to increase research in underrepresented biomes and clades. As Agnes said, for me as a Colombian was quite shocking and frustrating to see that Colombia doesn't have a lot of studies in melastomes, even that there are a lot. And if we think of the high Andes, there were also not that much studies. So we do really need to start including underrepresented biomes and clades in our study. It should be said, however, that our review was only recovered study, peer review papers and review papers. So there should be a lot of information in other kinds of sources, in research being carried out at universities and so on. But we weren't able to recover that in our review. Uh, if you remember that graph where I talked about the different stages of the germination ecology, you should remember that germination, the effect of abiotic factors on germination was by far the most studied topic. So we have a huge gap, huge gap in the loop between dispersal germination and establishment. We have a really, let's say, a good idea of how seed respond to different abiotic factors, but what happens before and after germination is something that we don't have that much of an idea. So if we really want to understand the seed biology of this group, from the moment that seeds are dispersed to the moment that they grow up and become a seedling, we should start doing more studies in other topics rather than only lab experiments about germination. And as I said, our approaches to compare the effect of abiotic factors on germination have been more qualitative rather than quantitatively. So recent studies are starting to use models, thermal time models, and more modeling approaches to do a more formal approach to compare the germination requirements of different species. So we should incorporate this sort of models in our studies in melastome if we want to understand the comparisons, for example, between growth forms, between different biomes and so on. We do need a more formal and statistical approach to do such comparisons. And in my whole talk, we were talking always about comparing different species and different biomes. But one thing that has not been studied that much is the interspecific variability of germination biology of this group. As you know, germination by as most reproductive traits, they exhibit a huge variation, for example, between populations located in different sites or between different years with contrasting climatic conditions. And that is something that we haven't studied that much on melastomes. So that's another gap that we should be thinking on. And finally, this is a huge problem in seed functional ecology as a whole, is that we are always thinking about germination traits and how they relate to easy traits like seed mass or seed size. So you will always find in the book chapter, for example, that we always make, seeds of melastomes require light because they are small and have a small size and so on, so on. And they are form a persistent seed bank because they are small, and we always go back to the seed mass and the small seed size. And we don't know how these responses could be related to other characteristics of the seeds. Maybe their internal morphology has something to say, maybe their dispersal phenology or something like that. And we should start doing more research in that sense. So as Agnes and I talk, all of I have presented is way more explained and way more developed in the book chapter. So if you are interested in the germination ecology of Neotropical Melastomataceae and haven't read our book chapter, maybe you can read that. Or if you are interested or have some questions, or if you want to read more profoundly what we wrote, there is the book chapter. If you are unable to access the chapter, feel free to email me or ask for the PDF on ResearchGate. We will be happy to share with you our book chapter. So that's what I wanted to share with you. Thanks you so much for your attention, and I'll be happy to take any questions or resolve any doubts.
Thank you so much, Carlos, for a wonderful presentation. I learned a lot. That was great.
Great.
Thank you very much. Are there any questions from the audience?
I have tons.
I also have many. I have no idea how seeds work really,
So
but then Fabian, you go ahead.
okay. One of the things that that very early works talked a lot about frugivory, about how birds consume seeds, and I just saw a paper a week or two ago about how God passage did increase germination in species in the Andes. Has that really been studied much? Because given that half the species have berries and are most of them bird dispersed or at least vertebrate dispersed, it would be interesting to understand what's happening there.
Yeah.
And then a second question you can answer later is like when you were talking about dormancy, what is like the longest you because those seeds have so little endosperm. I don't know how long they can survive in the ground without dying. So what is the longest that it seems that these seeds would be viable?
So, Fabian, thank you for your questions. Regarding the first one, yeah, theory suggests that gut passage by birds or any other mammals, other animals, sorry, it should be beneficial for seeds because fruits usually contain substances or hormones that prevent germination inside the fruit, since in the most cases of melastome we don't want germination to occur inside the fruit. There are a lot of compounds that prevent germination inside fruits. And for instance, there is a study here in the Campo Rupestre that tried to do an experiment regarding that. In the lab, they prepare an extract from the fruits. They produce an extract with the fruit. They macerated, that was the word I was looking for. They macerated the fruits and produced an extract and applied it to the seeds. And they found that when you put this extract in the seeds, they tend to germinate less, probably because fruits have some substances that prevent germination. So yeah, gut passage by birds or principally birds, yeah, in melastomes should be beneficial for seeds, but yeah, we do have a lot of studies in seed dispersion ecology in melastomes, and they usually show that the biggest problem that we have studying this is because of the huge amount of empty and non-viable seeds. Because you see that something is germinating, but you see a lot of things that are not germinating, but probably it's because they don't have no embryo at all. So that's one of the biggest difficulties with working in this. But in theory, and some of the experiments that have been carried here show that gut passage do increase the germination percentage of species. And regarding your second question, it's really interesting about thinking about the dormancy because we don't know. I mean, we do know an idea that some species have dormancy, and we were able to prove that in the lab that you apply for those that don't work with germination. For physiological dormancy, what we usually do is to apply gibberellic acid, which is the hormone that promotes germination. We see that when we apply gibberellic acid to the seeds, they do germinate much better. The thing is that we really don't know how these things work in the lab, but we don't know really much about how physiological dormancy is being broken in the field. Theory suggests that physiological dormancy is broken by exposure to periods either of high temperatures or low temperatures. So seeds are in the soil is kind of moist, and some physiological mechanisms are being activated, not so much to germinate, but at least something to start broken germination. And after a couple months in those conditions, physiological dormancy should be broken. But the thing is that I'm not aware of a field study that have tried to do this in the field. We only have like the theory of how physiological dormancy is broken.
I'll follow up with the question I had because I think it fits well. I really liked when at the end you came to talking about how you should or we should close the gap of dispersal, germination, establishment. And I was just trying to think of a seed bank and like I'm like shit. Okay, you said we should see if the seeds fall down and what happens then. Well, first, you need to get rid of the soil seed bank. Whatever. All of that aside, I know too little about all of this, and I just wondered whether you might share some insights or what you're doing for your PhD now to close these gaps. I'm just super curious.
So yeah, in my PhD, I'm not working only with melastome, but melastomes are part of the experiments. We are doing, and it's pretty related to what I just said. We have a really good idea of how dormancy is broken in the lab, but we don't know how this is happening in the field. So what we are doing is we are doing everything. We are doing an experiment in the lab and also an experiment in the field where uh, we are taking soil cores, soil samples, and germinating them in a greenhouse. So we can have a general idea of how the seed bank dynamic is occurring in the field. And we have some species that we are taking some samples of them and bury them in the field, and we are going to monitor them for a year to see if they are able to persist for a whole year or not, but also how or at which moment physiological dormancy is being broken. So we will have the natural soil seed bank dynamic, we will have the field experiment that will tell us in which moment dormancy is being broken or not in the field. And we also be doing a germination experiment in the lab that is called a move along experiment. In a move along experiment, you have your germination chamber, and you try to reproduce the temperature conditions that it should find in the field. But also, and you try to move them along and the conditions that it should have in the field. So, for example, in our case, we made it quite simple. We have the temperatures that it should find in the early dry season when these seeds are germinating, and we will move them to the late dry season and so on. But you also have a treatment when you put these seeds directly in the conditions of the rainy season when we expect them to be germinating. So if you take the seeds and they do not germinate in the condition of the rainy seasons immediately after you put them is because there is some dormancy mechanism that prevents them to germinate in that moment. But in the move along experiment, if the seed is able to germinate after passing by the temperatures of the early dry season, the late dry season, and then they germinate, you are able to say that dormancy was broken in this period of the dry season, at some moment of the dry season. So we should be able to, theoretically, we will be able to match the soy seed bank natural dynamic with the field experiment and the conditions in the lab to try to understand what is happening actually in the field.
Very cool, thank you. Are there more questions from the audience?
You can also, if you don't want to, you can't. They have a microphone or anything. You can also put them on the chat, if you want. Carlos, I gonna take advantage of the little pause and ask. So, there seems to be a huge difference in seed size um, across groups. And I was wondering if in the percentage of either presence of embryo or viability, that there is a difference. So like things with bigger seeds, because there are fewer seeds per fruit, they tend to have embryos because you don't need as much pollen. I mean, is there any correlation there?
The general theory suggests, as taking plants as a whole, is that bigger seeds usually are viable and have embryos because as a allocation thing, they have less seeds to feed, so they end up being bigger and having larger embryos. But regarding melastomes, we are not able to do such an association, and thank you for that question, because most of the studies that we have are with light seeded species, for species with really small size. So when we are thinking about trying to include underrepresented clades, we do really need to start doing germination biology of these melastomes of large seeds, such as Mauriti and these kinds of other species that have big seeds. Probably there should be a relationship regarding seed size and seed field, but with the data that we have so far, I don't know, especially because we have a lot of data from Campo Rupestre, and Campo Rupestre have all this problem about nutrients and pollen limitations. So yeah, unfortunately, at the moment, we don't have that information.
It was also really exciting what you said about how they need very short periods of light exposure to germinate. So could we imagine, now I'm thinking of a wind dispersed seed, I'm thinking of a Mariania in a cloud forest. So basically the seed flies, I don't even know how far they fly, probably not very far, and lands, I don't know, on a tree trunk with a lot of moss. Could the seed basically germinate right away if it is exposed to light?
In theory, yes,
Wow.
because yeah, in theory, yes, and this is a pretty common behavior when we are thinking about pioneer species, because they have a small size. So actually, they don't have, as Fabian pointed out, they don't have that many
Nitrogen to
reserves
sperm,
and
yeah.
nutrients inside them. So they do need to germinate in light conditions. So they start to do photosynthesis as quickly as they can. So yeah, in theory, we should germinate seed stability. If there is light, they should germinate right away. In the experiments that I've been doing so far, we have two melastome species, one pleroma and one ketostoma. And the pleroma, in less than 2 weeks, all seeds are already germinated. It's really, really quick. So yeah, I think in the case of most melastomes, if they don't have dormancy, they should germinate almost right away.
Wow, that's cool.
Okay, I have a question that might be too specific. So, do you have any idea about the success rate of the seed germination? of Orthostemma. Hello?
Sorry, I kind of lost the last part of the question. Could you repeat that, please?
Yeah, so do you have any idea about the success rate of seed germination of Orthostemma? It's
No.
one genus in the nootropics.
No, unfortunately, no. Unfortunately,
Okay.
by
Maybe
head,
Lou, you can work
no.
together with Carlos and try to germinate some in the greenhouse.
Yeah, good
Yeah, that
idea,
would
but
be
it's not really my project. Just someone from the lab is going to work on the genus. So yeah, just out of curiosity.
The more
Thanks.
data, the better.
yeah.
Sure.
I I also have a question, but I think I don't know much about seeds first or the seed, germination. And I just wanted to thank you. There was a really nice explanation as seed germination as a whole. And about also the light condition that you're mentioning to germination. I'm thinking about how mostly um melastomes are present on humid forests, because right now you're studying like in an open habitats. It makes sense, they can germinate in light, but thinking about humid forests as a more shadow environment, and how they are more present on this humid forest. And I was thinking about, so the intensity of light, does it change? How does that work?
That is a really interesting question. Thank you for that. And what we have to think, we need to think of something. If you are, let's imagine that we are a small seed in the soil. If you are quite small, you don't have that many nutrients to grow, to build a seedling and everything like that. So what this light requirement does is for you to avoid germinating pretty low in the soil profile. So if you are a really tiny seed, you don't want to germinate in the soil profile, because you will need to grow a great distance before reaching the surface. And if you have seen melastome seeds that are tiny, really, really tiny. So this light requirement prevents them to do this germination that won't result in establishment. But we have to think also that these seeds are able to persist for a long, long, long time in the soil. So if in forests, this is quite clear, if seeds are able to persist for a long time in the soil, but thinking about also the comment that Agnes made about light requirements, you are a small seed in the dark conditions, you won't germinate. But let's think that a tree falls and a gap is open. If there is light, you will germinate right away. You are going to let's use really well this new environment that has been created, and you are able to germinate right away and take advantage of those conditions. So this is probably why many, many pioneer species, not only melastomes, but Cecropia, Ficus, and many other pioneer species have this behavior. Of course, if you are regarding that, let me just go back to that slide that shows that result. You should be seeing my screen, and you should be seeing this table.
Yeah.
In this particular study, we are not talking about light intensity, but of course, light intensity should be an important factor, but we don't have that many studies about that. But regarding the light quality, uh, you see that, for example, the species in the understory, in this case, the ones in the green square, they need light, but the quality of the light that they need to germinate in the understory is not that of a much quality. They only need a small amount of red light to be able to germinate. But if, for example, Clidemia viscehata, Nosaea confertiflora, there are these other species that are from the gaps, and they won't be able to germinate in the understory because they require very much no, they need more red light, these values in the yellow square, they do need more red light to germinate. So even if there is some sort of light in the because you go to the understory and it's not dark, there is some light and so on, but the quality of that light, it's not enough for them to germinate. So yeah, we are thinking also in the composition of this light that is rich in the seeds. Depending on the microhabitat, this should be a limitation of whether they are germinating or not.
Thank you. Thank you so much.
It's already 11:03, and I know some people are starting to drop. Probably they have um, other things going on. Thank you so much, Carlos. Thank you, Agnes, mostly for re-engaging everybody on this.
It was you
And
in the summer.
yeah, and then Agnes has now lined up everybody for this between now and March, I believe. And
I
we
think
have a
that's true.
couple.
Yep.
Yeah, so we have December, January, February, March talks already lined up. Um, once again, if you want to volunteer, write to any of us, Agnes, Lucas, me, Vinny, and we'll slot you in, and then we'll go from there. So we'll see you in a month.
Bye, and thanks a lot, Carlos. Thanks.
Thank
Bye,
you.
everyone.
Bye.